Afternoon, everyone, and welcome back from lunch, and welcome to session 2, which is on emerging measurements. And this afternoon, we're going to hear about wildfire smoke and aeroallergens. And we have a slight change in the schedule. Our aeroallergen talk is on the schedule to be first, but due to some technical glitches, we're going to start with Katie Benedict from the Los Alamos National Laboratory.
So I'm going to talk about some new work that we've been doing to characterize bioaerosol emissions from wildfires, both in the field and in the lab. So just to get us all on the same page, this is a little bit broader than the aeroallergens sometimes this group thinks about. And so we can have different types of biological material in aerosols that really span the range of aerosols we traditionally measure in the fine and the coarse mode. These can include things like proteins, viruses, bacteria, spores, and pollen. And you might wonder why we're thinking about wildfire smoke. So some previous research has observed some differences between background, ambient measured bioaerosol, and smoke-impacted bioaerosol. So in this study, they essentially took a petri dish, put it outside, and they saw what they got. And then they tried to grow it, right? So they saw enhancements in the bacterial colony-forming units during different phases of combustion and to a lesser extent when different types of material were burned. I was like, well, I think we can do better than sticking a petri dish outside. Most of us in this room could probably, who are familiar with sampling aerosol. And so we sort of proposed a study, but since, in the meantime, this group out of the University of Idaho who did these initial measurements has also done some characterization of what these biological materials might be, right? And so they did some 16S sequencing to look at the different types of bacteria, archaea, and fungi, for instance, and they saw all of these different species, and we see just generally there are differences between the ambient and smoke-impacted sample, suggesting that the wildland fire smoke alters the composition, diversity, and the potential implications of these bioaerosol in the environment. I proposed that we look at how the smoke-related bioaerosol are different from ambient bioaerosol by doing both experiments in the lab where we control the combustion conditions and also in the field where we would get maybe more natural representation of what's going on. So this schematic sort of describes that. So we know that this previous study looked at the bioaerosols in smoke. We didn't know if that's directly from what they were burned or if they were from the lofting of soil that has a lot of microbes in it that was associated with some of that fire from winds. So in this study, we're going to use two different characterization techniques of the bioaerosol. They include a real-time fluorescence measurement as well as filter samples to characterize the viability or if the biological material is alive, and also do some DNA sequencing to better understand the impacts. So our first measurement, the real-time fluorescence measurement, is the wideband integrated bioaerosol sensor, or the WIBS. There are several groups around the country and world who have this instrument. And it's a single particle measurement of bioaerosol that takes advantage of autofluorescence of biological material. This instrument gives us the number concentration, the size, and an asymmetry factor, which is essentially a proxy for shape. There's fluorescence excitation at 2 different wavelengths and detection at 2 different bands. And these were chosen based on what we know about autofluorescence of biological materials. There's this great classification scheme that's a little bit intense, where for every excitation wavelength and paired detection wavelength, which there's only 3 detection bands because one of those gets saturated, we can identify if it's a singly fluorescent, so that gets a single letter. If it's doubly fluorescent, it gets 2. And if it fluoresces at all 3, it gets an ABC. The specifics of that aren't necessarily critical because we can't really identify, what that biological species is by this fluorescence. And previous work has sort of looked at this, and in this study, they aerosolized very specific single type of organisms, so bacteria, pollen, and fungal spores. And we see that even for a single type of species, you get a multifluorescent spectrum, essentially. But size is really an important factor here, right? The bacteria are all small, the fungal spores are all mostly big. But we see, like, for instance, type A fluorescence is very prominent in both the bacteria and the fungal spores, right? So it gives us maybe a little bit of an idea of what it is, but the size is probably more important. And overall, we're looking just to see if it fluoresces. In our lab, we have a tube furnace to do our controlled combustion, and so we preheat that to our set temperature on the digital display in the schematic. It's 1,000 degrees C. We introduce dry, particle-free air into that. We slide our sample in on a quartz boat. You can sort of see that in the schematic as well. At the exit of the tube furnace, we measure the change in oxygen, so how much oxygen was consumed during the fire, which is a good indicator of the combustion conditions, but we also measure carbon dioxide and carbon monoxide using a Picaro. And so we dilute that smoke that we generate from the tube furnace because it's so concentrated, we overwhelm our instruments if we don't. And then in the case of the cascade impactor, we need a greater flow than the 4 liters per minute that we run through the tube furnace. We need closer to 25 liters per minute. So we have a significant dilution factor here as well, but for biological material, we're not really concerned about changes associated with like gas aerosol partitioning. So here's an example of our 3 replicate burns at each temperature, 500 degrees C to represent smoldering conditions and 1,000 degrees C to represent flaming for a ponderosa pine fuel sample. On the left, we see our change in oxygen. So the red is the 1,000 degrees C. We see that's a really quick burning phase with a large drawdown in oxygen. The 500 degrees C is a little bit longer and slower combustion, which is not too surprising based on what we know about combustion processes. On the right, we have the total aerosol concentration in black and the bioaerosol concentration in green on the right side of that for the y-axis. And we see that, the production of aerosol closely mimics, the change in oxygen over the course of the burn. And we see really good repeatability in our replicate samples as well. Overall, we see that the fluorescent bioaerosol particles are 2 to 10% of all of the aerosol. And they sort of represent a similar fraction for both the 500 and 1,000 degrees C burns. But if we look a little bit more closely at this, if we compare the bioaerosol emissions for the different temperatures and the different fuels, that's what's shown on the left here. And this is an average of the 3 replicates we did for the juniper, pinon, and ponderosa pine samples, so all evergreens that we would find in northern New Mexico. We see that there are these emissions are more different for the temperature than for the fuel types. Right, we see some differences in, between the fuels, but 1,000 degrees C is much more obviously different than 500 degrees C. If we look at the fractional contribution on the right of the different type of fluorescent aerosols, we also see that those are pretty different between the temperatures. At 500 degrees C, we see more of the ABC type particles, while A dominates at 1,000 degrees C, with other differences also in those bars too. If we take a closer look at the smoldering combustion, instead of looking at the different, here we're plotting, I'm looking at the different size bins across the bottom x-axis. So these are binned according to the cascade impactor that we used, and I'll show you some data from next. So the smallest size bin is 0.5 to 1.1 micron, up to greater than 7 microns. And we see that for the smoldering combustion, the smallest particles dominate the emission, potentially suggesting these are more like bacteria rather than pollen or fungal spores. We also, again, see that they're pretty similar across the fuel types. There are some minor differences where, for instance, the juniper has a much larger fraction of A-type fluorescence than the pinon and ponderosa at the smallest size bin. But again, the fluorescence type is really hard to interpret at this point. If we look at the flaming combustion, we see a few more differences between the different fuel types. Again, we see that the most particles are emitted have the smallest size range, the 0.5 to 1.1 micron. But we see, for instance, ponderosa pine has a lot of B-type fluorescent biological particles, potentially. And juniper has maybe a little bit more diversity of the AB and the AC type particles that we haven't really seen when we looked at it in the other frame. So I mentioned the filter collection. We are using an Anderson cascade impactor. We're really interested in connecting potential impacts of the bioaerosols we measure to a variety of different outcomes, whether those are human health or ecological effects. And so the Anderson cascade impactor was sort of designed with the human respiratory system in mind. We use milk-blocked cellulose filters, so essentially we put a layer of milk that gets dried, and that helps keep the particles from penetrating into the filter, which we found was a sort of a problem for some of these biological particles. We've done some fluorescence microscopy. I'm not going to show that, and we have some DNA analysis planned, but today I'm primarily going to talk about flow cytometry. And so this is a method where we can stain cells using different types of fluorescence indicators, depending on what we're interested in, and then we can look at those and count them based on their fluorescent properties. So it doesn't take advantage of autofluorescence, it's an added fluorescence, and we're using two different stains in this work. The first is Cyto9 plus propydium iodide, which labels the cells that do not have a damaged membrane. So it should suggest that they are intact and potentially and likely to be viable. We also use FUN1, which identifies cells that are metabolically active by processing that stain and changing the fluorescence signal. So we're looking for a change in fluorescence signal for those. And here is a summary of those results. We have both stains for each of the columns and both combustion temperatures. The fuels that have no data, I'm waiting on the data for those. And we have a few more fuel types than I presented in the last few slides. We've added sagebrush and gamble oak and blue grama, which is a grass. So one thing I neglected to mention with those stains, they're not mutually exclusive. We're likely have some overlapping population of the two. But we can see that those stains have different, sort of indicate different viable cells in each of the stages and for each of the fuels so that they're not completely, overlapping or mutually exclusive. One interesting thing here is we saw in the WIBS data that we saw more fluorescent biological particles at 500 degrees C. Compared to 1,000 degrees C. But if we focus on the pine needle data for the Cyto9 stain, for instance, we see that there are more viable cells produced at 1,000 degrees C than 500 degrees C. So that's a little bit interesting. We also see that, where those cells are in the size range changes a lot. And we see, many more particles in this sampling that are larger than like 4.7 micron compared to what we saw in the WIBS, where we saw the smallest particles dominate. That could be a function of our sampling setup and how our lines are for the WIBS sampling, and it's something we're still exploring. And then we went and we did some prescribed burns at the Kanza Prairie National Grasslands. And so we took our same sampling setup, put it in the back of an expedition, and we added some size distribution measurements, and also we took a microaethalometer so we could make sure that we could filter the data by when we were in smoke. I sent a student and two students and a postdoc. And just to show you, we burned plots that were small and big, and it's just really flat grassland in this area. And so if we look at this prescribed fire data, we took some upwind samples to get the background, and we also, here I'm just showing burns from the small plots, but the data from the small and the big plots are pretty similar. Similar to what we saw in the lab, we see more more particles in the smallest size bin. For the burns, we also see those in the natural background ambient, but we do see an enhancement in the fire compared to the background site. We also see, if we look at the fractional contribution of the fluorescent types, that it's pretty different between the two types of samples, suggesting something different is happening and being produced during the plume than at the upwind sites. If we also look at the flow cytometry results, this is just the one Cyto9 stain. We don't have the FUN1 stain yet. We see elevated viable particles during the prescribed burns. And here I'm averaging the day 1 and day 2 samples together for both the smoke and upwind periods. And we see on day 1 a significant enhancement in the total number of viable particles, quite a few viable cells, particularly in the 2.3 to 4.7 size range. nope, I read that wrong, 2.1 to 3.3, and also 4.7 to 7 size range. We didn't see the same degree of enhancement on day 2, but we do see some enhancement, but definitely not the enrichment we saw in those 2 size bins. So just to summarize, the number and type of bioaerosol vary with the fuel and the temperature. Temperature is definitely a more important factor. We saw the most particles in the field and in the lab in the smallest size bin. And so the work that we're planning on doing next is to look at how soil production near these fires might impact the bioaerosol concentrations. We have samples of the soil and the grasses from the Kanza Prairie, so we can try to tease apart, exactly what is contributing to the bioaerosol there. We are in the process, I believe, hopefully the lab has them now, doing some DNA sequencing of the viable fraction to better characterize the implications, right? And so I mentioned, there could be human health implications if these are pathogenic or even impacts to crops and ecosystems downwind as these biological particles that maybe weren't originally from there get deposited in these places, particularly with the increase in fires that we've seen related to climate. And then we have another field campaign to go do some prescribed fire measurements in Georgia this February. And so this will better be better to compare with some of the more evergreen type fuels that we burned already. And so unfortunately, the project's going a little bit slow, and so I can't really talk about what the impacts of the deposited smoke are, but hopefully that is something I could come back in the future when we have those data and talk about as well. So thank you.
Any questions for Katie?
Hi, great talk. So do the, these bioaerosols, their behavior and characters change with the photochemical aging process?
Yeah, so probably yes. And we actually have an oxidation flow reactor in our lab where we're going to try to better understand how the aging potentially would change the properties and behavior and our ability to detect them. We're still kind of a long ways from that because we've been having so much trouble with our biological analysis, but some other people have shown, maybe not necessarily directly related to smoke, but other bioaerosol measurements that the properties do change with photochemical aging.
Really interesting, Katie. I'm trying to think about what the source is of these particles. Like you mentioned, maybe sometimes you're getting the fire fronts lofting soil and doing things. Has anybody done experiments where they pass individual bioaerosol types through a flame front to see what happens? I mean,
so not to my knowledge. If I can get permission at Lanel to aerosolize some biological particles, I think that would be really interesting to do. But in our samples where we burn them in the lab, there's no soil. There's just pine needles.
Yeah, it's really interesting. And I can imagine that maybe some things survive and have fragments that fluoresce, but maybe aren't viable, that could be different. But there's a lot of interesting experiments to do, I guess.
Yes, I agree.
Thanks.
Thank you, Katie.
I think they might be doing one more question. You can cut them off.
Really interesting talk. So just a couple of questions about the fluorescence analysis. So you were saying that it's difficult to assign chemical meaning to the fluorescent categories that you've identified, right?
Yeah.
But I was wondering, what are the real benefits of that? Are you using those different classifications, A, C, to compare against literature, or is it just something that a way for you to categorize the different fluorescent species and then kind of build up on that and compare it to your own data or compare it to the literature?
Yeah, primarily it's a comparison to literature at this point. I actually think we're going to move away. Like when I publish this work, we're probably going to present it, but we're not going to talk about it as much. One of the interesting things that I didn't present was more about the shape factor, right? So the shape factor that the WIBS gives us can tell us whether it's long and rod-like, whether it's cubic, or whether it's spherical. So it's like, sort of an estimate that might provide actually better information about what it is rather than the fluorescence type. And presenting the combination of the shape with the size, I think is the way forward. But right now, people who have a WIBS are presenting fluorescence types, so I am too. Great, thank you.
And our next speaker is Ross Edwards from the University of Wisconsin. And there you go, Ross.
Hi everyone, and good afternoon. So today I'm going to talk about this project of opportunity that we conducted just recently in the Great Lakes with, in collaboration with Ryan Lepak, the EPA. And so obviously, if you lived in the northern part of the eastern US, you would have been aware of the massive fires from Canada this past summer and the smoke and air quality problems. And so a lot of that smoke was passing over the Great Lakes. And so we sort of wondered, we've been measuring black carbon in snow and rain and a while back in some lakes, and so we wondered whether we could be able to see the black carbon deposited to the lake. So this, sorry. Oh, okay. Got it. Okay. And so just quickly though, just to acknowledge that this study would not have been possible without support from the EPA and the staff and of the RV Lake Guardian, which is the EPA's research vessel, and also staff from the Madison Water Science and Engineering Laboratory. So the impetus for the study, so as I said, we had these large smoke plumes passing over the Great Lakes, and there's a lot of reasons that we're interested in these plumes. Personally, I'm interested in black carbon aerosols, so little refractory nanoparticles of carbon, little onions, and which affect the climate, affects cloud structure, cloud formation, precip. But also these fires are rapidly redistributing nutrients and toxic species, whether they be metals, etc. And they impact terrestrial and aquatic and marine ecosystems when it's deposited. But if you're looking for traces of wildfire, of fire emissions, black carbon is pretty close to an ideal tracer. It's refractory. It doesn't degrade. There's only one source, which is incomplete combustion. And these particles are also interesting in water bodies because they may also take up species like PFAS or chemical species that don't normally mix well with water, can pass into these black carbon nanoparticles which have really high surface areas, and then be transported into the water that way. So there are a lot of reasons to be interested, and also we're also interested in the processes which preserve black carbon in the sediments for looking at the paleo history of deposition. So we were able to get samples from the RV Lake Guardian summer survey. So this was conducted during August and sampled all the Great Lakes. And so this was my first interaction with this survey, but there's a survey that's conducted in the spring and then in the summer, but it supports the EPA's water quality monitoring program in the Great Lakes and the biology monitoring. And this was, I think, the 40th summer survey, but we were able to get a total of 181 samples. You can see the picture of the vessel, but also the rosette up here that was sampling down through the water column. So we just got samples from near the very surface of the lake, so in the top 5 meters, let's say, and then, or top 2 meters, and then at the bottom of the lake, to whatever depth that was, which, so like, I think it's 2 meters from the bottom. And the samples were collected directly from the flasks on the rosette into the vials. So we had little vials, and personnel were able to just basically squirt it directly into the vials. And you can sort of see all these sites in this map. We sampled a lot of these sites, but there may be a few that are different. And so just to rehash the black carbon story, some people here have heard this many times now. But so yeah, black carbon is the product of incomplete combustion. It's initially hydrophobic, so it doesn't really interact with water typically, but it's transported in the atmosphere, becomes functionalized, reacts with nitric acid, and gets coated with things like sulfate, and becomes, as it ages, it becomes hydrophilic, let's say. These particles, they're really like little semi-fractal grapes, bundles, but the actual spherules may be about 30 nanometers in diameter, the little round spheres. And then a typical black carbon particle that's transported in the atmosphere may have I don't know, like a mobility size of, say, anywhere between 200 nanometers, 300 nanometers. So it's fairly small. And then the main pathway out of the atmosphere is in rain, so it's in wet deposition, both above and below clouds. And the stuff that we actually measure is what we call refractory black carbon. So that's these little carbon onions, which can be heated. They can reach a temperature of about 4,000 Kelvin before they incandesce. So there's very few materials that can do that. But then what happens when it gets deposited to water bodies? There's a number of factors that control the stability of black carbon in water bodies. So these aged particles are initially stay suspended by electrostatic repulsion, so they can stay suspended in water samples for a long time, unless the ionic strength or the pH is incompatible. So high ionic strength will shrink the charge cloud around these particles, and then they can aggregate and they can bind onto other things. If they get coated in humic acids, they could stay suspended for a very long time. And in this case, the particles are stabilized in the water column by what's called steric stabilization. There's an entropic stabilization. These particles, the coat of humic acids, could essentially transport down the ocean, down rivers into the ocean. And there's a large body of research that's being conducted to understand long-term carbon cycles, which are looking at black carbon from fire and how it's found in the ocean sediments throughout the world, and there's questions about how it gets there, how it actually transports. But so for our work, not sure that little, so I've managed to activate some kind of little submenu. What's that? Just click again. Escape? Or will the whole thing go away? All right, there we go. So, so black carbon is not only being transported, deposited to say the Great Lakes from the air. It's also come from the sediments, because that's where it's all ending up. There can be some resuspension that releases it from the bottom. Also from the nearshore zone. So there's let's say runoff and river inputs, where if the black carbon's stable, then it can come in. Or even in the case of runoff from the city, you might expect that there's huge amounts of black carbon actually pouring off the sidewalk and off roads in stormwater and going into the lake that way. And then, so there's sediments near the shoreline as well. So, and the main process is probably heteroaggregation with other suspended particles, where the black carbon will basically aggregate with those particles and be rapidly transported to the sediments. And also biological materials and plankton and stuff like that it will stick to. So we're analysing these black carbon by the most sensitive way to analyse these particles is by laser-induced, intracavity laser-induced incandescence. So essentially we're heating up the nanoparticles in the heart of a laser system and to the point where they glow. And that's happening around 4,000 Kelvin, and we have a way to detect the temperature. And so we know that one of these little particle, these incandescent events is black carbon by its temperature. So there's very few things that will, like I said, get to 4,000 Kelvin. Iron particles will be, let's say, 2,000 Kelvin or something like that. So we have a way to differentiate between black carbon particles and other particles. But in terms of water, what we're pumping water into this system, but first we're passing it through a desolvation system. So we're pumping water at about 450 microliters per minute into like a little heated section, a hot zone, if you will, with a carrier gas. And then we're removing the water, and then we're condensing the water off the particles, so we're left with a dry aerosol stream, and we're passing that into this incandescent system. And the limit of detection for this is about 30 parts per trillion. So it's very sensitive, it's highly linear. It's linear over many, many orders of magnitude. But what we are doing is we're getting particles within a certain size range. And to us, that's really mass. So it's between 0.3 and 80 femtograms per particle. And so we're detecting every particle, and then we're summing up that mass of those particles over a certain time period. So in this case, every 5 seconds. It gives us an aerosol concentration. And then we have standards, which are fullerene soot, dispersed fullerene soot standards, which behaves very similarly to the black carbon in the atmosphere, and we're using those standards to calibrate the system. Okay, so these are the results. This is what we found. So this is the surface waters from the Great Lakes. And so a lot of the samples were, let's say, only about 22% of the surface samples were above 100 parts per trillion, very low concentrations in general. But there were places where we found actually some large surface, what I would call large surface concentrations. So Lake Erie, off Toledo, I guess, and where the concentration was about 700 parts per trillion. Lake Michigan, we had some sites where we had a maximum of 500 parts per trillion. And then in Lake Superior, it was a sort of a wider, not as high concentrations in the surface, but the spatially higher concentrations over the surface. But there were plenty of places where these concentrations were so low, they're lower than Antarctic snow, incredibly low concentrations. So by and large, we found very low concentrations, but there were episodes which may be atmospheric deposition. So this is the surface waters, but if we look at the near the bottom, so we found I would say less at the bottom, but there were some sites there was one place in Lake Superior and or two places really where there was some higher concentrations near the bottom, and this may be from turbidity, from sediments coming up. And then the other place was really Lake Erie again, near Toledo. Whatever's happening down near Toledo and Detroit, and it's producing fairly high concentrations. And in that site, we think it's probably from the cities, right? It's probably not. It's hard to show that it's coming from the atmosphere, but it could be runoff from the cities. But also, it's a shallower lake as well, much shallower lake, and so we saw some higher concentrations there as well. And just to compare these, I don't know how I can I go backwards? No. It's all right. I'm going to escape out of that. So those concentrations were in parts per trillion. This is a previous study that we had published published in Environmental Science and Technology, and this was with regards to the fire in Lake Tahoe in 2007, the Angora fire, where the ash was literally landing on the lake. And so we did this, we were able to get repeated profiles from the lake. And we could see a rise in the black carbon in the lake just after the fire, but then rapidly decay. And so the concentrations we found from the deposition to the lake were pretty similar to what I just showed you. So we found concentrations up to like about 400 parts per trillion. These are in this little box here, I guess the lake profile that's in parts per billion, but in parts per trillion it's about 200, 400, 500. We also looked at runoff into Lake Tahoe, and there we found in different types of rain events, especially snow, we found concentrations up to 600 parts per billion, so almost a part per million in some cases, from runoff from roads and things like that. So you can imagine, this is Lake Tahoe, it's a pretty small watershed, but you can imagine somewhere like Chicago or... okay. And just to speed up, so this is, we also did this is just a pilot study from Crater Lake, the biggest bucket in America. Has essentially no watershed, it's just the surface area of the lake is the watershed, and it's the deepest lake in the US. And so you can see, it's kind of tantalizing, but you can see higher concentrations at the surface, around nearly 200 parts per trillion again, not, this is in parts per billion. And then this profile dropping down through the lake, which is consistent with deposition to the surface and then particles just moving through the lake. If you could if you knew enough about the hydrology, you could invert this to calculate a deposition rate. But similar, the 200 parts per trillion, so in comparison, the maximum that we saw in the Great Lakes is almost 800 parts per trillion. Okay, not sure if I'm being fast or slow, but so just the conclusions. This is the first study of its kind. No, this is the first time anyone's done this, so we're looking at the data. This is preliminary data, but it does look like that we could see some differences between the different lakes, and we could see some events which looked to be atmospheric deposition from fire smoke from Canada. And the interesting thing to me is Lake Erie. The inputs to the parts of Lake Erie could still be fire-related, but it does look awfully like black carbon coming from the cities around the southwestern end. I think the follow-up of this is that there's more work to be done, obviously, but this is going to keep happening, I would say, right? The fires in Canada, these big fires, we don't expect any less in the future. So whatever comes with that smoke in terms of nutrients, metals, whatever, so we can imagine this is going to increase. So it's another area of research I think we need to continue. But also in close to the cities, there's this, we talk about the urban gradient. I think the urban gradient in these lakes must be pretty extreme, something like black carbon. Anyway, that's it. Thank you.
Thank you. So sometimes when there's smoke or black carbon deposition into a lake, it triggers algal blooms, and sometimes there's deposition and it stops algal blooms. Do you think that could be related to kind of the chemistry happening around the black carbon, or do you think it's too complicated a system to?
No, I think there's in that case, there's usually some limit. I don't know about the inhibition, but there's limiting nutrients. So there's something else coming in with the black carbon. It's probably phosphate or nitrate, I would imagine. But the inhibition depends how much. I guess if you have You know, I'm not sure what it would mean for algae, but I've read reports of fire emissions triggering algal blooms, not just in lakes, but in the ocean as well. There's a couple examples in the southern Pacific, for instance. In that case, it was metals. It's probably iron, bioavailable iron.
I guess I have a quick one. What's the comparison to black carbon measured within like the NTN samplers?
Oh yeah, sorry, I forgot to mention that. No, that's a very good question. So the concentration ranges, concentrations that we saw in the Great Lakes was obviously less than 1 part per billion, to the point of being, you know, basically not below a detection limit of 30 parts per trillion. But in the rain samples, the concentrations that we have seen in the rain samples, we have seen a few very low concentrations, maybe half a ppb, Bermuda, for example, but typically anywhere between a couple of parts per billion to 10 parts per billion, but in some of these big events, up to 70 parts per billion. So it's a pretty big range, but typically between 5 and 10 parts per billion would be kind of normal. So, but orders of magnitude higher. So, and I guess like the PFOS question is one I keep thinking about, because again, it's a question whether if you have 50 parts per billion in rain, can that also would that affect PFOS concentration in rain? Which I have no idea.
Thanks. Good talk, interesting stuff. So you're interested in seeing if there's a connection between atmospheric deposition of black carbon to what you're seeing in these lakes. And you mentioned there may be some connection based off of what you see in the surface water samples.
Yep.
And you might have already talked about that, or maybe I missed it, but did you look to see if there's an agreement between what you see at the surface water samples and plume data? See, like modeled? Yes?
Sorry. Yeah, not yet. Not
Okay.
yet. So yeah, that's the next. This is really recent. This data we just recently got out. And we're waiting for the rest, the other data that's from the cruises. But yeah, then we'll look at all the rest of the chemistry as well.
Okay. Cool. Thanks.
Okay, thank you, Ross. And I'm hoping our next speaker is Chris Lawrence from the University of Albany. Okay, take it away, Chris.
All right, good afternoon, everybody. My name is Chris Lawrence. I'm a PhD candidate at the University of Albany at the Atmospheric Sciences Research Center in Albany, New York. And today I'm going to be talking about, I think, something a lot of us are pretty well aware of after this past summer, the impacts of transported wildfire smoke, specifically on the Whiteface Mountain chemical system, and more specifically, cloud water chemistry. And just a couple of acknowledgements early on. I want to thank University of Albany and the Atmospheric Sciences Research Center for helping, supporting me through my degree, the Adirondack Lake Survey Corp and the Adirondack Watershed Institute, who are really important in analyzing the chemical data I'm going to be showing you today, NYSERDA for funding the cloud water collection program, and then NASA for funding my, my graduate fellowship. So I don't think I have to spend too long talking about wildfire is becoming a pretty significant air quality problem throughout North America. And of course, there's a lot of fears that they're only going to get worse over, as we undergo anthropogenic climate change. And there's already some evidence emerging that air quality progress that's been made in the United States is starting to stall or get worse. This figure here is from a paper published in Nature just this year looking at PM 2.5 trends across the country, and their results found that the vast majority of states either saw stagnations in their improvements in air quality or actually saw reversals, PM 2.5 concentrations getting worse. So there's already evidence now that wildfire smoke is degrading or reversing any progress that we've made. And 2023 was a banner year for wildfires, particularly in the Northeast United States, a region that is just not accustomed to being impacted quite like the West Coast is. And these were largely driven by fires in Quebec, the province of Quebec, that burned from late May into really late into July. And it put, especially during the week of June 6th, give or take, it put much of the Northeast under really severe air quality warnings with air quality indexes over 200 for several hours, with cities like Syracuse and Rochester and New York City really getting hammered. And that's not even talking about the poor cities in Canada like Ottawa that really just saw the brunt of this. And just in general, in more recent years, if you look at PM 2.5 trends in a city like Albany, New York, you can see in more recent years, the ones I have circled in red here, that PM 2.5 trends have shown spikes above regulatory standards. The bottom dotted line on this plot is the 12 micrograms per cubic meter EPA standard, and the upper dotted line is that 35 microgram 20 per cubic meter 24-hour standard. And you can see in recent years that we've exceeded both of those standards quite frequently in Albany, New York. Again, a region that's not typically used to wildfire influence, or at least aggressive wildfire influence. So the motivation of this talk is that we know that smoke events can lead to really significant air quality problems. However, the chemical composition of wildfires is still less understood, and there's a lot of uncertainties. And as it relates to cloud and wet deposition, that is even less known. So I'm going to be talking about some preliminary work that I've been doing for the past few months, investigating actual impacts of wildfires on cloud water chemical composition at Whiteface Mountain. So if you're unfamiliar with the site, Whiteface Mountain is in the Adirondack Mountains in upstate New York, and it's home to a really historic cloud water monitoring program that began in 1994 with the major goal of monitoring the progress of the Clean Air Act amendments in the 1990s, really looking at acid deposition. This is a fantastic site for cloud water monitoring because it has a really easy access to the summit during the summer that has electricity. And we're in cloud anywhere between 20 to 60% of the time, allowing for frequent sampling. And just important to note that this monitoring only happens between June and September, so these are only the summertime months. Overview of the collection system. It's an automated system, a MONI omnidirectional passive cloud water collector that just deploys Teflon strings when certain meteorological parameters are met, including detection of cloud, above freezing conditions, enough wind, and no precipitation detected. So these are non-precipitating clouds that we're measuring. And the Teflon strings have water beat up on them, and they move through an array of tubing into a refrigerator accumulator, which then dumps these days every 12 hours. So the time resolution on these cloud samples is every 12 hours per sample. So unfortunately, I couldn't bring any cloud water data from this past summer for you. We do have it, but we're working very hard to analyze it. But a researcher in Albany, Dr. Jia Zhang, was smart enough to actually deploy our aerosol mass spectrometer during the really nasty fire event that hit New York from June 3rd to June 11th. And the results are on this plot here showing the PM1 mass. And you can see that the vast majority of the aerosol composition, greater than 95% of it, is organic in nature. It's organic matter with a little bits of increases in nitrate as well. And we can actually use this data to actually help constrain what we would expect in our cloud water chemistry. And indeed, if you look at other biomass burning events that impacted Whiteface Mountain, include, for instance, one that occurred on July 20th, 2021, that was largely influenced by prairie fires in Manitoba, Canada, you can actually see that TOC concentrations, or total organic carbon, spike during these wildfire events. In this particular event, upwards of 4,000 micromoles per liter of total organic carbon, which crushed any record we have at Whiteface Mountain. And 2021 had the highest median concentrations of total organic carbon, coinciding with a year with a lot of wildfire influence. And this is all happening in the backdrop of an increasing trend in median total organic carbon concentrations at Whiteface Mountain. And the reasons behind this are still pretty unknown to us, but we do have some evidence now that wildfire smoke could be contributing. So I really wanted to investigate, can we actually quantify the contributions of wildfire to this total organic carbon trend? So first, you could start off with something like a multilinear regression, comparing inorganic ions we measure in the system to total organic carbon. So this is just some model results I showed, and this multilinear regression model does pretty well, has an adjusted R-squared of about 0.7, so getting about 70% of that variability. And I use standardized regression coefficients, which allows you to actually compare the individual regression coefficients more apples to apples, since they have different concentrations. And the one I have circled here is potassium, which is often thought of as a useful biomass burning tracer. And it does have a positive relationship to total organic carbon. It might be a little hard to see, but you'll note that the regression coefficient here is actually the smallest of the other ones, which kind of indicates that while there is a positive influence from potassium, it's limited compared to, say, a dust tracer like calcium, or maybe an anthropogenic tracer or agriculture tracer of something like ammonium. But multilinear regression is notorious for being difficult to actually figure out what causality is rather than just correlation. And it's possible that this potassium tracer might not just be coming from wildfires, it could be coming from dust. While I did try to remove that from the regression model, it still could be there. So this has some limited applications. So I wanted a better way to actually quantify smoke events and smoky days versus non-smoky days. So what I used was this fantastic data product from NOAA, the NOAA Hazard Mapping System, which is a satellite data product that actually just shows you where smoke events are occurring, and it can actually give you some information on the density of that smoke. And I used a very, very simple algorithm here. If the NOAA Hazard Mapping System had detected smoke over Whiteface Mountain, I considered that a smoky day versus a non-smoky day, a very simple algorithm. So this is an example of the type of information you can get from the NOAA Hazard Map System, where this orange shading represents a heavy density of smoke over Whiteface Mountain on July 20th, 2021. And this works very well. We know that was a very smoke-influenced day. So this is a proof of concept of this, using this data in determining smoky days. So to start, right now, I'm just comparing the total organic carbon concentrations on days that had smoke detected versus non-smoky days. And the median TOC concentrations were nearly double on smoky days compared to non-smoky days. So this algorithm does seem to detect a noticeable signal in the total organic carbon concentrations that is statistically significant. We can actually start looking at things in terms of month to month, with July typically being the most commonly smoke-influenced day of the, or month of the year, with just about a third of days in July being influenced by smoke, and August also being quite significantly influenced. And again, if you split it up month by month, you do see a statistically significant difference on smoky days versus non-smoky days. So this does seem to be able to detect a signal. And we can also look at the trends in smoky days over time at Whiteface Mountain. And I really want to point to the bottom right plot here, where there does seem to be an increasing trend in smoky days occurring at Whiteface Mountain, with up to 90% of the days during the summertime collection season in 2023 seeing smoke influence, which is, I think, a pretty incredible result. And here I split up the median total organic carbon concentrations by smoke influenced days and non-smoke influenced days. And again, you can see that the smoky days typically have higher median concentrations of total organic carbon, but aren't seeing any noticeable trend. But if you look at non-smoky days, they have a lower total organic carbon concentrations, but they still continue to see that increasing trend in TOC. So the way I'm interpreting this is that some of the increasing trend of total organic carbon can be explained by wildfire smoke, but there's a significant fraction of this trend that cannot be. And it's an open question on what's actually causing this. It could be maybe increased biogenic VOC chemistry contributing to this, but that is an active area of research for our group right now. And I just want to leave you with this. This is regression, it's just some simple regression of total organic carbon versus some important inorganic ions that we measure in the system for smoky days versus non-smoky days. And you might have a little hard time seeing the actual regression coefficients, but you'll notice that the regression coefficients really aren't very different. They're very similar values. The slope of the regression line just isn't very different, though you do see that the R-squared values do go down, so the model is not quite as predictive. But I think the most interesting part about this is that if you look at the intercepts rather than the regression coefficient, so the way you would interpret an intercept in this case would be if you set your ion to 0, this is how much total organic carbon you have in your sample. And the intercept is about double, if not more, for just about every inorganic ion that you do this regression analysis on. So the way I'm interpreting this, though, it's, if someone has a disagreement, please let me know, that the relationship between total organic carbon and inorganic ions in the system doesn't change on wildfire days and non-wildfire days, but the base amount of total organic carbon that you have is typically higher. And this is kind of a surprising result because it's pretty well known that fires release a lot of other things besides organic carbon, especially a lot of nitrogen. So I'm a little surprised to see that the relationship with other inorganic ions is actually not very different for smoky days versus non-smoky days. So this is something we're going to be looking into more detail and has some pretty important potential implications on how increasing wildfire influence could be affecting atmospheric deposition processes in the future. Just some brief conclusions. Wildfire smoke, as I hope to convince you, is becoming a growing problem throughout North America, and it can lead to pretty significant spikes in total organic carbon concentrations. And that the preliminary NOAA hazard mapping data analysis that I've done shows that TOC concentrations are nearly double on smoky days versus non-smoky days. And that there's evidence that the number of wildfire days impacting Whiteface Mountain are increasing and can explain some, but not all, of the increasing total organic carbon trend. And finally, there's no obvious change in how TOC interacts with other inorganic ions in the system, which complicates our understanding of how wildfires are going to affect other things that this network is really interested in, such as nitrogen. Happy to take any questions.
Hi, thanks for the talk. Really interesting. It's going to be really great to see your cloud water samples once you have those results too. Two things. One caution about using the HMS as an indicator of smoke is that means there's smoke somewhere in the column.
Yep.
And we see, like in the Midwest, we see, basically the whole summer now we seem to have smoke somewhere in the column, but it only reaches the ground a small fraction of the time. So we have it like we actually have been combining ground, looking at kind of deviation of PM2.5, carbon monoxide, and ozone from the mean, and then combining that with HMS smoke present. So that's sort of one way we've been using to kind of try to, and you kind of get a combined metric. We're using a bunch of different parameters to try to figure out if you have smoke at ground level. So
we should definitely talk later about that.
Yeah, I'm happy to talk about that.
Yeah, you're absolutely right that this is a very simple approach that I took here, and it's very limited. It's the column, not the actual concentrations at the surface. I was very surprised to see how strong that signal actually was. It not just like a little bit difference, nearly double. So this is just very preliminary work. So any advice you have on that, I'd love to hear it.
Yeah, happy to talk about that. Also curious, I'm guessing since you're focused on the aerosols, you didn't, did you look at gases at all?
We've done a little bit of VOC chemistry, but not a whole lot. We're very aerosol and cloud water chemistry right now.
Okay.
Yeah.
Just curious. It's something we're this summer we saw a lot of. We had like 7 or 8 episodes where it got to the ground here, and we saw a lot of ozone enhancement. So we're trying to figure out: is it the NOx? Is it VOCs? What's coming in the plumes?
It's complicated.
It's complicated. Yeah. Yeah. Thanks.
Have you tried using the HMS or any other kind of fire mapping product to look at effect of other wildfires? So maybe not directly over the mountain, but if your dominant wind direction is coming from the north, is there wildfires in Canada that would explain that extra
That is
TOC?
absolutely a next step. Right now, I only had a couple months to put this sort of stuff together. So that is the next step, trying to get more upwind transport, not just the smoke that's over Whiteface Mountain. And there have been some people who've done something kind of similar to it, and we're going to look into probably try to use their methodology because it's a little bit more robust than the current methodology that I'm using right now.
And then second question, I guess, have you tried estimating TOC emissions using that HMS or like any sort of product like that?
I have not yet. If you have any suggestions on how to approach that, I'd be very interested.
I know how to do particulate emissions, but maybe not TOC as much. But
It would probably still be helpful information.
Great talk. I was really interested in the your cloud collection device. How much water what's the kind of amount of water do you get out of that?
It's variable, of course, but in a 12-hour period, we've seen up to like 10 liters of water. That's not every day, but it's over a 12-hour period, a few liters is pretty common.
Okay.
Yeah. All right, thank you very much.
Thank you, Chris, that was great. And our next presenter is Terry Williams from the Wisconsin State Lab of Hygiene. And, oops. All right, Mike, where is the swap? Upside down doesn't work.
No.
There we go. Thank you.
Okay. So my name is Terry Williams, and I'm a microbiologist at the Bioaerosols unit at the Wisconsin Occupational Health Lab, which is a part of the State Lab of Hygiene and under the umbrella of the School of Medicine and Public Health here at the UW-Madison. So I do have to apologize. I had a co-presenter here today who was going to set up half of what he and I did working on a project for NADP. And so I'm going to stumble through most of what he was going to talk about and then jump into my half of the presentation. So normally what I do in the bioaerosols unit is analyzing samples for fungi and bacteria, usually for indoor air quality investigations. So including in that, we do a lot of Legionella, and because of some of that, our air samples, we do get a lot of pollen in there. As you can see here, we have our fungi, Legionella, some pollen on top, and we always, because it's air samples, we're going to get the occasional parasitology in there. So it's a cute little fairy wasp, I thought I'd add. So because we have a total spore analysis that we do, which is impacting spores onto a gel strip to count spores, we get a lot of pollen in there, and we're able to count pollen. So I was approached by some of the NADP members who were collecting all this data. So NADP collects rainwater, air filters, everything, and they were wondering, what else can we use these samples for? And pollen was brought up, and so I was approached in helping do a study on comparing different ways to count pollen from environmental samples. So because I'm still going over my co-presenters, so if you can see, I wrote some notes here, so if I get that deer in the headlight looks, I'm just trying to decipher my handwriting. So, so the study that, or excuse me, the paper that we published out of that, and I'm going to read this because it does not fly off the tongue easily, is initial comparison of pollen counting methods using precipitation and ambient air samples and automated artificial intelligence to support national monitoring objectives. So included in this paper, we had a couple of people working on it, including Greg Weatherby, David Gay, who is up here today, and Eric Urim, who was supposed to be presenting with me. So if I get stuck on anything, I'm totally throwing David under the bus, and we'll ask him to help me answer some questions, myself and Andy Johnson. So what we did was, using the samples that NADP already collects, including the rainwater, and we collected that by the NADP wet-dry collector. Everybody's familiar with what that looks like? I apologize, I don't have any pictures of it, but we used the wet-dry collector to collect rainwater samples that we would filter and count pollen from there. The other type of samples that we had were air samples. So it would use a high volume pump, and it would send the air through an air filter. A lot of NADP sites already collect these air filters, and we thought we could also count pollen from there. And that's a yes and a no at the same time, and I'll get to that later. The other way that we collected samples was through, or that we assessed data was through a pollen sense, which is an automated artificial intelligence that it would take a pump that was sending air through a jet, and that jet would impact any spores or any particulate that was in the air onto a revolving tape. So if you imagine a tape, like a reel-to-reel kind of recording device, it's continuously monitoring, and as it's going through this jet and impacting on that tape, the AI would make an identification as to what type of pollen was impacted on there. So then you would be able to get counts through that, which is great because since we have that tape, you can always go back to look at that. Those were the types of samples collected. Before I really get started, just want to say that if I happen to mention any specific names or products, that it's just what we use, and it's not an endorsement by the lab or the university. It's just what we're using, so blanket statement. When we first started figuring everything out, we knew that we had filters. NADP was already using some of the filters, and you can see the opaque filter on the lower right of the screen. We had wanted to use that, but when we put it under the scope, we weren't getting enough light to come through, so we had to kind of figure that out. What would allow us still to see the spores or see the pollen grains well enough, and we had David had looked up a filter from the Steritech Corporation, and they actually made a filter that was see-through, and it was pretty much night and day when we look at what it would show us. So in the upper left was the opaque filter with the polyester sulfone, is what I believe NADP currently uses, and what we found were the polyester filters. We had to be conscious of what pore size that we were using, because we didn't want to lose any particulate to go through the filter, so we chose a 0.5 micron pore filter. These are about 47 millimeters in diameter, so maybe about the size that a pop can would make if you see the ring when you set it down. So that's about the size of the filter that we're looking at. So the pore size is about 5 micron, but when these filters were put through the high volume pump, it created a lot of back pressure, and it really messed everything up for the pump. So we figured out, well, we can't use those. So Dave went back to the drawing board. We found a 3-micron pump, or 3-micron pore size. And when those were used in the pumps, we were able to get a good airflow to go through. So we had a good collection efficiency with that. So you can see, having a larger pore size really didn't mess up what we were trying to look at. When we identify pollen and spores, we need to have enough resolution to be able to see certain markers. So having a clear surface to look through, a clear membrane, was very important. Now the 3-micron initially was a concern, but I think the smallest size pollen is about 6 microns. So we're pretty happy with the fact that we weren't going to miss any pollen, and we were still able to get a good airflow that went through the pump. This is the setup that we had at the lab. Like I said, when we were receiving air filters, we would have to put them on a slide with stain and cover slip them when they were ready to read. The rain samples that we received, we would put the same type of filter that we used in the extraction that you can see in the upper left. So it's just a reservoir with a magnet that you're able to switch out. We put the filter through there and filter about 250 milliliters of rainwater. This ended up being a good volume because otherwise, if we had a lot through there, we would end up overloading the filter and we wouldn't be able to see through it. So the other step that we had to figure out was we knew we wanted to use the Calburla stain, which was good for pollen, but we didn't exactly know when we have, a 47 millimeter or in diameter filter, how are we going to drop stain on there and get nice even coverage? And turns out the easiest thing was to just put it in the water. So as we're setting up the filter and running the water through, we would just add the stain directly to that water, and as it filtered, stained it perfectly. It ended up being really good. Then we were wondering, well, when we received just filters from our sites, how are we going to stain those? And we ended up having to do the same thing. We would just get sterile water and run them through the same process. So now that we were able to set up those slides, here's what we're looking at under the scope. So we wanted to be able to compare counts, but we didn't really have a really good way to count them. So when we're looking at a 47 millimeter filter, that's actually a lot of surface that you have to go through when you're counting something. So if you, for instance, if you're looking at a football field and everybody just threw their sports equipment, all the kids in high school just, here's footballs, baseballs, shoes, everything, and we're walking off the field. Now you have to inventory all of that equipment on the field. But you can only count what you can see on the yard line that you're walking down. So it would take forever to count that entire football field to get everything counted for. So that's kind of what we face here. When we put something that big under the slide, it's like counting a football field. So how do we get past that to get counts? Well, one thing that we'd have to do is just do a random sample. Similar to something we do for like asbestos, like the NIOSH 7400 method, we would count at least 10% of the sample, which is not great. Of course, we want to count 100%, but 10% gives us a good starting point. Maybe it introduces issues with reproducibility or issues on, you know, are we missing something? Yes, true, but that's a very big field to count, so we have to narrow it down. Otherwise, we would be counting all day. So what we ended up doing is we start at a random spot, and we'd count through everything. We would identify pollen grains, fungal spores, and we'd write it all down, move to the next random spot. Do another identification, count, tally those, move to next spots. And if we did 176 fields of view, that equals to about 10% of the sample. To help us out in recording all that, here's the counting sheets that we would use. Because of, like I said, our experience with total spores, where we see pollen all the time, it's pretty easy to make that crossover into counting the pollen. Calculations actually ended up being the easy part, which that doesn't always happen. But all we're doing is taking our raw count, divide by the number of fields that we counted, and then times by the entire area of the slide. So, and then your units are just a calculation as per your units. So what let's see, I had some other notes. So when we did crunch the data, we had some good correlation between total pollen counts and tree pollen. The grass and weed counts were a little more variable, but I think overall we had a good start on a couple different novel ways to actually count pollen using the resources that NADP already has. So, questions?
Not a question, but I just wanted to put this out there to the folks joining us on Zoom. If we don't get to an online question or you have a hand raised, you can put your question in the Q&A section of Zoom and we can get to it that way.
Good.
I guess first off, I guess I'm more interested in, like my line of work is like the species that are responding to deposition and that might be changing across the country. And looking at this, like I saw in that last table, you had a couple of tree genre at least that you had clearly classified out. Like, can you identify common weed allergen species through this?
Yes. So when we first started, like I said, we had seen a lot of pollen come through. When we first started this, we had seen a lot of pollen come through, so it was a good intro for us to actually start learning how to characterize those. In the paper, they did get pushed into, like, this is tree pollen, this is weeds, flowers, that kind of thing. But yeah, you can very much distinguish between, oak, ash, and actually get into genus. The spores that or the pollen grains that you see on the slides have all of the characteristics that you need to do that. So yes. Mm-hmm.
I was just going to ask you about how often in the wet deposition samples were the pollen grains lysed or broken apart versus whole?
That happens either way. And we didn't know, was that an effect of, the pollen grain was sitting there. They are hydrophobic, so I don't know if that had anything to do with them lysing, but I don't have a count as to, we still count anything that is crushed or crumpled or already lysed, but I don't have those statistics up against each other. It's always a handful of any type of species that you'd see, but I don't have a good, maybe 10%, maybe, but once they get that crumpled, it's often hard to tell what type they are. So they would just get set into their own category of miscellaneous or broken. Mm-hmm.
And I guess one other thing, can you differentiate like microplastics, or did you see many microplastics on the slides?
That did come up. I haven't been instructed or figured out what actually that looks like. I know we do some particle ID, and I wanted to check that out. I think with the pollen sense, they are able to, they're picking that up a little bit more, but I'd have to check. Do you know, David, do they? Mm-hmm. So, and I know that's an emerging concern at the moment. So, yeah, pollen sense does have that in there, but I don't know if they publish the data or not. Mm-hmm.
Great. Well, let's give one last round of applause to all the speakers in this session.